Hi everyone. So today I'll be reviewing the article that developed an isolation method of IgG antibodies from combinatorial libraries. So the current cell surface display faces some challenges such as screening issues. Even with state-of-the-art instrumentation, screening libraries with diversity greater than 10/8 is very challenging. Also, the process is very biased towards antibodies that are not very best at binding to the target, not to mention inefficient translocation of potentially useful antibodies. So to overcome the problems, the work developed a technique where antibodies are expressed in the cytoplasm of E. coli. A chimeric antigen is created by fusing the antigen to an antibiotic enzyme CAT. The chimeric antigen is designed to be transported out of the cell through the TAT pathway. However, if antigen binds to the antibody, this cannot happen. So the enzyme can inhibit the antibiotic activity and the bacteria can survive. Next, they wanted to test if the presence of an antibody that binds to the target can protect bacteria from the antibiotic. So they focused on this CDRH2 region of this anti-GCN4 cyclonal antibodies. Bacterial grown can be observed in the absence of antibiotic. However, with antibiotics, only bacteria with stronger antibody variants survived, and weaker binding variants show very little to no growth. This plot here shows the binding strength of the antibodies using ELISA. The stronger binding variants had higher binding affinity. On the other hand, the weaker binding variants showed lower binding activity. Next, in order to improve the binding affinity of the targeted antibody, a combinatorial library was designed based on the variant with lowest binding affinity as seen in the previous slide. So different variants of GFA with different sequence in CDRH2 region was created, as this region is known to be important for binding strength. And all the variants here showed good binding for the target. Other targets here were used as a control to test if the antibodies are specific. And all this data here suggests that initial variant did not bind well, but the other variants did, showing that mutation in CDR region affected binding. Next, the researchers tested their new antibody selection method to target an influenza antigen. For this purpose, they constructed a fully human cyclonal library comprised of heavy and light chain sequences derived from transgenic mice genetically engineered with humanized IgG. They were able to find two variants, 20.2 and 75.1, that can strongly bind to the antigen. Specificity results here show that the variants are very specific for the target, and little to no binding was observed for the BSA antigen. And the KD value of these variants were very decent considering that they came from a non-immune library, meaning that they were not designed against the influenza antigen. So the study developed a genetic selection method for isolation of antibodies from combinatorial libraries expressed in the cytoplasm of E. coli, and it is possible to screen important antibody variants using this approach, and those selected variants showed very decent binding even after humanization from non-immune library, showing that this is an effective method for antibody selection, and also it addresses some of the limitations of cell surface display discussed earlier. Thank you so much for listening, and I would love to answer any questions.